Hello, my dear students. Welcome to my channel. Here we'll study 12th Standard Biology. Lesson number 2, Reproduction in Lower and Higher Animals, Part 5. Now, in this lesson, we'll study about the gametogenesis. What is the meaning of gametogenesis? The meaning of gametogenesis is the process of formation of gametes in sexually reproducing animals means it is a process where the gametes will form either the spermatogenesis or either the oogenesis. These two sex gametes will form, and these gametes will contribute in the reproduction in the animals or in the humans. The male gamete is called the sperm and the female gamete is ovum or egg. The gametes are formed from primordial germ cell of gonads. Okay. So in this process the will also form and the gametes will also form. So this process is called as the gametogenesis process. So this process divides into two parts, the spermatogenesis and the oogenesis. In spermatogenesis, the sperm formation of male sperm will takes place, and in oogenesis the formation of the female gametes, that is ovum or oogonia or egg formation will takes place. Spermatogenesis is a process where the formation of male gametes or sperm or spermatozoa from the germinal epithelium of testis is called the spermatogenesis. At the onset of puberty, the hypothalamus begins secretion of gonadotropin, releasing hormone. It initiates the significant increase in the secretion of follicle stimulating hormone, which includes spermatogenesis. Each seminiferous tubule is lined by a single layer of cuboidal epithelium cell called germinal epithelium. The cell of germinal epithelium undergoes spermatogenesis to produce sperms. The process of spermatogenesis involves three phases. The first one is the multiplication phase. The primordial germ cell of seminiferous tubule undergoes repeated mitotic division to produce large number of spermatogonia. Each spermatogonium is diploid and with 46 chromosomes. The next one is the growth phase. Some of spermatogonia stops dividing and grow in size to develop into primary spermatocytes, which is two in number due to accumulation of food. Now the third is the maturation phase. It involves mitotic or reproduction division. The spermatocytes undergoes the first phase of mitotic division, that is mitosis first, leading to formation of two haploid cell called secondary spermatocytes, which are with 23 chromosomes each. The secondary spermatocytes undergoes second phase of mitotic division. Mitosis second, the to produce four haploid spermatids. The spermatids is non-motile and non-functional. It gets transferred into functional spermatozoa by the process called spermatogenesis. During this process of change, the spermatids remains held to each other and to sertoli cells by cytoplasmic bridges. The sperm head remain attached to the steroli cells and their tail hanging in the lumen of seminiferous tubules during spermatogenesis. The length of spermatid increases. Centrioles are reached as primary and distal centrioles. Mitochondria becomes usually colloid and acrosomes is formed from Golgi complex. Now we'll see here the structure of male gametocytes or the sperm. It is divided into three parts: head, tail, and the middle piece. The sperm is a male gamete. It is a motile, microscopic, elongated cell. It is divided into three parts: head, middle piece, and the tail. As you can see in the figure, head. The first one is the head. The sperm head is oval in shape and contains haploid nucleus. Above the nucleus, there is a cap like structure called acrosome. It is formed from the Golgi body. Acrosomes contains hydrolytic enzymes. The hydro Nized and proteolytic enzyme like zona lysine and corona penetrating enzyme. The next one is neck. It is a very short region having two centrioles, that is proximal centriole and distal centriole. The next one is the middle piece. It has an axial filament surrounded by 10 to 14 spiral turns of mitochondria, that is Nebenkirin. It produces energy necessary for the movement of sperm. The next is tail. It is a long cylinder and tapering part containing cytoplasm and fine thread axial filament. The axial filament arises from the distal centriole and travels throughout the length of tail. It is partially surrounded by plasma membrane or main piece. The part without plasma membrane is called end piece. Now, as you can observe in the figure, the three main parts of the sperm is there. The head, it contains the acrosome, nucleus, proximal centriole. Okay, and the middle piece, it contains the neck, and the tail is used as the motile part. It is help to movement of the sperm throughout the vaginal cavity. Oogenesis. This is a process of formation of the female egg, that is oogonia or eggs. Now we'll see here, as you can see in the figure, the detailed structure. The process of formation of haploid female gametes, that is, egg or ovum, from the diploid germinal epithelium. It involves the process of meiosis and mitosis. Like spermatogenesis, oogenesis process can be divided into three stages. The first one is the multiplication phase, the second is the growth phase, and the third is the maturation phase. So the first one we'll study the multiplication phase. It is the stage the primary germinal cell, which is two in number of ovary, undergo repeated mitotic division to form millions of gametes, mother cells or oogonial cell. This process is completed in the embryonic stage of human female. The second one is the growth phase. Some of the oogonia stops division and begin to increase in size and form the primary oocyte, which is 2n in number. Cellular organelles like endoplasmic reticulum, Golgi apparatus, and mitochondria increase in the number. The third one is the maturation phase. Oogenesis takes place in the ovaries. The process is initiated prior to the birth of the female baby. The primary oocyte, two in number, enter the nutritional phase, which includes mitotic division. Meiosis first and meiosis second. The diploid primary oocyte undergoes meiosis first, that is reduction division, to form two haploid daughter cells. This division is particular in female as both the daughter cells are the haploid number of chromosomes, that is 23 chromosomes. But due to unequal division of cytoplasm of the two daughter cell produces, one is the large cell called secondary oocyte and another is the small cell called first polar body. Normally, the first polar body does not enter meiosis second. The secondary oocyte produces meiosis second only up to metaphase second. Its division is further stopped or arrested at this stage. The secondary oocyte is shed from the Graafian follicle and ovary. The restart and completion of meiosis second will happen only with entry of sperm. This last phase is usually complete in the ampulla of the fallopian tube at the time of fertilization. In this division, also, the two unequal daughter cells are formed. The last cell is ovum and the small cell is second polar body. The ovum is formed, functions as the female gamete and is ready for fertilization. Completion of meiosis second and completion of fertilization go hand in hand. If the secondary oocyte does not receive the sperm, the spermatozoa it is shed off along with the menstrual cycle. The structure of secondary oocyte or the structure of an ovum or unfertilized egg. In human, unfertilized egg when ovulated that is released from the ovary is actually the secondary oocyte. It is non-cleodic without shell and in microlethal yolk is present in very small quantity. It is approximately zero point 0.1 mm or 100 microns in size. It is rounded micromotile and haploid female gamete. The nucleus of the egg appears large and is called germinal vesicle. Typical nucleus or pronucleus is formed at the time of fertilization. The cytoplasm of egg is also called ooplasm. It is divided it is devoid of centrioles. The egg is surrounded by various coverings. As you can see in the figure, the egg membrane is called a veterinary membrane. It secretes a non-cellular glycoproteinous membrane, zona pellucida, on its outside. Adhering to the outer surface of zona pellucida are several rapidly elongated cells forming the corona radiata. These cells are derived from the innermost layer of granulosa cell. They are firmly held to the zona pellucida and to each other by hyaluronic acid. Between the vitelline membrane and the zona pellucida is a fluid-filled per- perivitelline space. The first polar body lies in this space. The egg shows polarity, the side having germinal vesicle and first polar body is called animal pole, while the side opposite to it is called vegetal pole. So here we have studied the detailed structure of sperm, the detailed structure of ovum, the process of spermatogenesis, the process of oogenesis. Okay, all the phases, all the stages with the diagram, with the structure and its function we have studied in detail here. Thanks for watching. I hope you like this video. Please like, share and subscribe the video and press on the bell icon to get notified for the every new video. As you know, we have taken the 100 days challenge, so till 100 days daily I will upload a one new video. So it is a humble request from my side. Please watch the full video without forwarding or skipping it and help me to complete the challenge of 4000 hours watch time. And please share as more as possible, as much as possible, because I want to complete the 1000 subscribers also. So this is a fifth part of lesson number 2, that is reproduction in lower and higher animals. This is a fifth part. In next part we will study about the fertilization and the other parts. Okay, so thanks for watching.